Alright, Unit 9 is going to make up most of the new stuff on your final. It's the last significant unit. I'm not even going to bother with a unit for Unit 10 because it's very simple. You guys will be fine on it. I don't even think you guys need this video. You should kind of have it down by now that you've gotten 3 Bracho tests under your belt. So we'll just go through this one nice and quickly. Make sure to know the functions of your lipids. And I have a chart in your notes for this unit that I made. I think someone revised it as well, so it looks even nicer for you guys. Really find that chart, find the linkages and where they're used and all that, the size of them. That's going to help you probably more than this video, to be honest. Find that summary chart. If you don't know what I'm talking about, just post about it, and we'll get a copy available to everyone. So 3 types of fats here. The omega number. We'll talk about the main types. We have omega-6s here and omega-3s. So you have one more saturation to make it an omega-3 fatty acid, giving it one more letter. One more letter here, linolenate versus linoleate. Make sure you know those. And definitely come up on a question that you know the difference of them. A few more good things to read and review. Palmitate, 16, oleate, 18. Whenever you see it like this, that would be no unsaturations. But if it has 1, that means there's one unsaturation in there. Don't worry too much about the numbering. Let's see, big ones to know here are these guys, linolenate and arachidonatate, which is 24. I sometimes think laurate might pop up or something for being the smallest one, so it's always good to know that. Palmitate is a pretty standard 16 carbon acid. I would know that one as well. But definitely don't worry about memorizing all this stuff for sure. Just focus on the big and the small. Know these points. Look at the questions for how he's going to ask about which one is going to be solid or liquid at room temperature, which one has the highest or lowest melting point. The patterns are pretty easy, pretty general chemistry on there. So know that tags are your storage. These are your 3 membranes. In your notes, just abbreviate these GPLs and sphingolipids. So this is an overview of what the rest of the unit looks like. So glycerol, there's 1 carbon here, 2 carbons here, and 3 carbons here. So glycerol is a 3-carbon backbone that's going to have all sorts of stuff. So for a glycerophospholipid, it has 2 fatty acids attached, plus a phosphate and an alcohol. This one comes up big in Biochem 2, so sometimes make a note of the structure of this one, having the 2 fatty acids plus a phosphate on the 3rd carbon. Let's see. These are some common alcohols, and the ones to know, I believe, tend to be choline and ethanolamine, because they're the ones that are going to give you positive charges. Those positive charges make it out to be neutral. So phosphatidylcholine here has a negative and a positive, so net charge of 0. Phosphatidylethanolamine has a negative and a positive, giving you a net charge of 0. You'll notice all the other ones have net negative charges here. So know these 2 as being neutral. Plasmalogens, anytime you see something like this, he's probably going to ask about it. There's only so many questions he can derive from this unit, so when you see something like an ether-linked alkene at carbon 1, you need to instantly go to plasmalogen. So cerebrosides, basically brain fat and sugar. Cerebrosides by themselves, basically a sphingoside with a sugar attached to it, a single sugar, glucose or galactose. If you have a couple sugars attached, 2, 3, or 4, it's going to be called a globoside. Remember, one sugar, glucose or galactose, is going to be a cerebroside. And then if you have multiple, it's going to be ganglioside. Out of this family of all of them, ceramide is the smallest, because it only has a little hydrogen attached to it. Again, I believe it's sphingosides that behave like fats, but they're not actually fats until you start adding other fatty acids to them. So it's kind of like the base product of all these fats. It's not a lipid itself. Cholesterol, know that it is a 27 carbon molecule for sure. Know that it has a little bit of polarity in ring A and an unsaturation in ring B. And it's a membrane lipid. So gangliosides, make sure to look up an old test how he asks about this. These 3 antigens come up on every test, so definitely look into how he's going to ask and what it looks like in test question format. So next part is the digestion of dietary lipids. It's good to read through this stuff. This looks familiar. Biosalts, this is a good example of one he might throw out. Help digest lipids. And this is a big chart to know as well. So if I was drawing this out, I would do it differently. I would put like for the first one here. Now they're all water dependent, so you're going to add water here, and then you're going to get, so you're reacting a triglyceride, which is most common, with water, and you have pancreatic lipase, and it works with something called colipase. That's what we're looking at here. What products are you going to get? You're going to get 2 free fatty acids, and then a monoacylglyceride. So the third fatty acid is still attached to that glycerol backbone, and it's attached to the second carbon. That's why there's a 2 there. So there's the full reaction. I think it's easier just to know in this way. So do that for the other 2 as well. If it's asked what the most common product of digesting dietary lipids, it's fatty acids. It's found in all of them. Cholesterol itself, if you just have regular cholesterol, it doesn't need an enzyme. You can just absorb it and use it if you need it. It's only cholesterol esters here that need to be broken down. Cholesterol esters look like it's just a cholesterol molecule and a fatty acid. So you take a cholesterol ester, add water to it, activating it with this enzyme over here, and you get a fatty acid plus cholesterol. Lipases help break off one fatty acid at a time. After you break them down to get them through the cell membrane, because only fatty acids or monoacylglycerides can go through, then they reform into triacylglycerides, and you add some other stuff, and they form chylomicrons. This is stuff you're going to learn about in biochem too, but FYI, it's things like HDL and LDL, those cholesterols everyone hears about. Big takeaway from this, this is another way of showing everything that we've looked at so far, that fatty acid synthetase. Remember synthetase, enzyme class 6, you have ATP there, so you activate it and then that's how you're reforming it And all of these different things go into the chylomicrons, like HDL, LDL. So it's made up of fat, phospholipids, cholesterol, cholesterol esters, and proteins. Activated with that synthetase, that ATP-dependent enzyme. Some questions you could ask on this one are coming from this column here, the composition. So know that the chylomicron is mostly triglycerides. I think I might have misspoke a little bit, like semantics, calling LDL and HDL chylomicrons. They're very similar to chylomicrons. The whole subfamily, or the whole superfamily is lipoproteins. So out of lipoproteins, these are the ones you need to know about. Chylomicrons themselves are mostly fat. VLDLs, these guys here, are mostly fat as well. About half fat, half other stuff. Intermediate, or IDLs, are going to be equal fat and cholesterol. Because to figure that out, that's fat. Cholesterol is a combination of cholesterol esters plus cholesterol. So it's those two combined. So it's 38 and 38. LDL, the supposed bad cholesterol, is mostly just cholesterol. Whereas HDL, the so-called good one, is mostly protein. Good amount of phospholipids in them too, but that's most likely what will be asked. Maybe that's the most common stuff. You might see something like which one is the largest on their own. So we have chylomicrons are very big. HDL is very small. While these are very big, they're also have a low density. He might ask about the smallest one, like chylomicrons have the lowest amount of cholesterol in them and the lowest protein, but that's tough. So I would definitely know the highest stuff first. So these are our big ones, these are our small ones like we just talked about. This is more digestion stuff. I think it's summarized on a chart here at the end. Yep. So this is what I will leave you with. Know this chart pretty well. It'll come up in a couple of questions. You won't need anything really study-wise for Unit 10. It's a pretty short, simple unit for nucleic acids, if you even get there. So good luck with your final and everything, and hope these videos helped you.